Hello, everyone. Welcome back to my video tutorial for single-cell RNA sequencing data analysis. Today, I'm going to show you how to use the Maxfuse package. This is a package recently reported on Nature Biotechnology. You can see they developed this method to integrate spatial and single-cell data across modalities with weakly linked features. So Maxfuse stands for matching X modality via fuzzy smooth embedding. So at the moment, they only have the package available in Python. Hopefully, we also develop the R package, so that will be good for R users. So in today's video, I just make a short introduction about this method. So you can see, firstly, use this method to integrate a protein and RNA dataset from a CITE-seq PBMC dataset. So in this dataset, there are 228 antibodies for the protein assay. So because of the high amount of proteins, they call it as strong linkage. You can see they use the Maxfuse to integrate the protein and the RNA data. And the cells from both assay were integrated very well. And at the same time, they used the thread package to integrate the same dataset. Because this dataset has 228 antibodies, the linkage will be strong between protein and RNA assay. So thread also integrated the data very well. But if they reduced the antibody numbers to 30, here they call it the weak linkage because the antibody numbers is low for the protein assay. Then you can see Maxfuse still integrated the protein and RNA data very well. But when the antibodies reduced to 30, then thread package cannot integrate the protein and RNA data very well. They also compared this method with other cross-modality data integration methods such as NIGA and Harmony, and also another method called bind-SC. I didn't show the image here. You can see Maxfuse is the best package to integrate the protein and the RNA data when the linkage is very weak. So after the CITE PBMC dataset demonstration, they also demonstrated the use other dataset for the Maxfuse package. For example, they integrated the RNA and the protein data from CITE-seq PBMC dataset. This dataset only has 25 antibodies, and they showed very good integration for the antibody sequencing dataset. This dataset has 97 antibodies, and also they show the data integration for protein and ATAC data for other technologies. For example, the ASAP-seq PBMC dataset. ASAP stands for ATAC with select antigen profiling sequencing. This dataset has 227 antibodies. And the fourth technology is the T-seq, which means transcription, epitopes, and accessibility sequencing method. And in this dataset, it has 46 antibodies. Like the ASAP-seq data, here they also integrated the protein data and the ATAC data. You can see from 4 different sets, the Maxfuse package can integrate the data very well for protein and RNA and protein ATAC-seq. So at the moment, there is no package can integrate codex data with RNA-seq data. We demonstrated in this paper using Maxfuse package. They can integrate codex data with RNA sequencing data. You can see in this dataset it has 46 antibodies, and they compared other methods such as Niger, binds C threat, and harmony. You can see MaxFuse performs much better than other packages for integrating codex data with RNA sequencing data. So after this video, we will use the codex and the RNA dataset as the example to show you how to integrate protein and RNA data using the MaxFuse package. They also produced the UMAP to compare the MaxFuse with Surat, Niger, Bind, SC, and Harmony. You can see probably Harmony is the best one here, but other methods couldn't integrate the protein and RNA data very well. So in this paper, they further used the MaxFuse package and performed the triple data integration. They demonstrated this package can be used to integrate the data codex, RNA, and the ATAC-seq data. So this is probably the first package to show you how to perform triple modalities data integration. In this dataset, they used the colon samples, and for the protein dataset, there are 47 antibodies. In the same paper, they also demonstrated the method for triple modalities integration for codex, RNA, and ATAC-seq data for small bowel cells. You can see here again, MaxFuse can integrate three datasets very well. So seems MaxFuse at the moment is the best package to perform cross modalities data integration, especially when the features link between different modalities is very weak. Then this package can perform data integration very well. That's the reason I'm going to make videos for this package. So that's my simple introduction for this package today. If you really want to understand the package, you can read the Nature Biotechnology paper. So I'm going to stop from here for this video. I hope to see you in my next video.